स्टूडेंट्स ऑफ क्लास 12 हम पढ़ रहे थे डीएनए फिंगरप्रिंटिंग। वही हमें कंटिन्यू भी करना है फिंगरप्रिंटिंग। मैंने आपको बताया है दैट द डीएनए फिंगरप्रिंटिंग हैज बीन इन्वेंटेड बाय अलेक्स जेफरीज said Alex Jeffries in 1984, and this DNA fingerprinting is a technique. It's a technique used in the identification of individuals. We can identify individuals. Identification of individuals on the basis of their respective DNA profiles. उनके DNA profiles हम देखते हैं तो हम इनको आइडेंटिफाई करते हैं। DNA profiles हम देखते हैं इंडिविजुअल्स के तो हम इनको आइडेंटिफाई करते हैं। एंड दिस टेक्निक used in the identification of individuals on the basis of their DNA, on the basis of their respective DNA profiles. It is known as DNA fingerprinting. And you know that there is 0.1% of DNA almost, which is different in all the individuals. This 0.1%, it contains VNTRs, जो मैंने आपको बताया है। VNTRs means sequences of DNA repeated so many times, and these VNTRs are known as variable number tandem repeats. Variable number tandem repeats. इन्हीं variable number tandem repeats को हम स्टडी करते हैं। They constitute about 0.1% of the DNA. They are different in all the individuals. They may have repetitive sequences repeated 100 to 1,000 times, in some 100 times, in some 200 times, in some 500 times, in some 1,000 times. The repetitions vary, and these repetitive sequences give us the identification of individuals. There may be small repetitions known as microsatellites, or there may be large known as mini satellites. Micro and many satellites. This DNA having the sequences, repetitive sequences, are known as satellite DNA. And the satellites may be microsatellites and mini satellites. Microsatellites are the satellites containing less number of repetitions, and mini satellites are those VNTRs having more number of repetitions. Now there's a procedure behind the DNA fingerprinting. First step is isolation of DNA. Isolation of DNA. We have to isolate DNA from a cell, for which we have to go for fingerprinting. This DNA is isolated from a cell. From animal cell, we can identify the organism. It may be taken from bacterial cell, we can see the bacteria. The first step is isolation of DNA. And the second step, this DNA isolated from the host cell, it is broken into small pieces, cutting of DNA, broken into small pieces, cut into small pieces using an enzyme. That enzyme is known as restriction endonuclease. Restriction endonuclease. With the help of the restriction endonuclease, we can cut the DNA into fragments, and the fragments obtained, they contain VNTRs. These fragments are later on in the third step multiplied, cloning. Multiplication of the desired fragments, cloning of the DNA fragments by means of the technique which is known as PCR, polymerase chain reaction. Polymerase chain reaction, PCR, इसके जरिए हम इनको मल्टीप्लाई करते हैं। और उसके बाद दूसरा स्टेप है, we can transfer these on a slab which is known as gel electrophoresis. The technique is known as gel electrophoresis, arranging the fragments according to their size and charge. Arranging the fragments on the basis of their charge and size, which is known as gel electrophoresis. Gel electrophoresis. With the help of gel electrophoresis, using a gel slab, the fragments can be arranged according to their charge and size. And later on, it is treated with alkaline chemicals. Alkaline chemicals so that the phosphate ends are removed. And after the treatment with alkaline chemicals, the another step is we have to go into the laboratory. We have to make special probes in the laboratory. The special probes should contain the variable number of tandem repeaters present on the gel slab, and we can prepare the slabs. We can prepare the we can say the probes. But before the preparation of the probes, we have to go to the other step, transferring. Transferring on a nitrocellulose sheet, nitrocellulose nylon sheet. We can transfer the DNA fragments from the gel slab on a nitrocellulose nylon sheet so that the film is obtained on the nitrocellulose nylon sheet, and then we can add probes. Prepared in the laboratory. These probes can automatically get attached with the VNTRs, and after they get attached with the VNTRs, we have to develop the X-ray film, which is known as autoradiography. Autoradiography में क्या हम करते हैं, हम एक्सरे फिल्म उठाते हैं इस नाइट्रोसेल्यूलोज शीट का, जहाँ पर ये VNTR बाइंड होते हैं with the probes prepared in the laboratory. Dark bands appear, and these dark bands show overlapping regions which are the DNA fingerprints. Again, I can tell you the steps. The first step is isolation of DNA. Second is cutting of DNA into segments by using restriction endonuclease. And the third step is cloning of the DNA fragments obtained by means of PCR. Another step is arranging the DNA segments on a gel slab by means of gel electrophoresis, and later on we can transfer these on a nitrocellulose nylon sheet. After transferring them on a nitrocellulose nylon sheet, we can make special probes in the laboratory and add these probes on the nitrocellulose nylon sheet so that they may get attached with a specific DNA segments, and special film is obtained by the process of autoradiography. X-ray film is obtained, and these overlapping regions are the DNA